In the late 1800s, the first clones were created by a man named Hans Driesch. Driesch's original goal was not to create identical species, but to prove that genetic material is not lost during cell division. Driesch used a sea urchin for his experiment because this animal has large embryo cells and can grow independently from their mothers. He took the embryo from the sea urchin and shook it in a bucket full of salt water until the two cells separated. After a few minutes, he looked at the split embryos closely and found that they had grown independently to form two separate whole sea urchins. Now let's travel ahead in time 102 years. It is the year 1902. Embryologist Hans Spemann used a piece of hair from his son to separate a two-celled embryo of a salamander, which, much like Hans Driesch's experiment, grew into its own separate salamander. Spemann did a few more tests on smaller salamander embryos, and then proposed what he called a fantastical experiment. His fantastical experiment was to remove the genetic material from an adult and use it to grow another adult. This was the first idea of human cloning.